Hello viewers, most of you must have observed people going for different types of exercises every day. But did you ever think what exactly is happening to the muscle in such cases? In fact, contraction of muscles and relaxation of muscles are taking place. What is this muscle contraction? In fact, that is our interest of this episode today. But before we go into the details of mechanism of muscle contraction, let us quickly recollect what is a muscle and how many types of muscles are there. Now, we all know that muscle is nothing but a bundle of elastic fibers which can be tightened or loosened to create some movements. And coming to the types of muscles, the muscles can be classified into three types. One is striated muscle, other one is non-striated muscle, the three is cardiac muscle. Now, striated muscle is also known as skeletal muscle or the voluntary muscle. Now, they have striations on the muscle, hence they are known as the striated muscles, and these are also called as skeletal muscles because this type of muscles you find associated with the skeletal system, and they are voluntary muscles because we can control the movement of these muscles, hence they are the voluntary muscles. Now, the examples are tricep and bicep muscles. Now, coming to the non-striated muscles, they are also known as the smooth muscles and they are involuntary muscles. That means the movement of these muscles cannot be controlled by the individual. We find these muscles lining the stomach, blood vessels, ureter, intestine, etc. And the third type of muscle, that is the cardiac muscle. The example is the heart muscle. Now, this is also involuntary. We all know that we cannot control the movement of heart muscle. So, this is involuntary muscle and this is intermediate in between the striated and unstriated muscle. Now, let us see the structure of the skeletal muscle. Now, as we say it is the skeletal muscle, we find it associated with the skeletal system. And in fact, these muscles, they form the major bulk of the human body. Now, in human body, we have 400 skeletal muscles. Now, all these muscles are supplied by somatic nerves. And not only that, each muscle will have a thread-like structure that is called the tendon, by which it is attached to the skeletal system. Now, if you just see the muscle, the entire muscle is covered by the connective tissue layer that is known as the epimysin, inside which you find many cells. Those cells are known as the myofibers. Now, these myofibers are also in turn covered by another connective tissue that is known as the endomyosin. These small myofibers, they come together to form another smaller bundles that are known as the fasciculus. Fasciculi is the plural and fasciculus. So, these fasciculi, they form the larger bundles such as the tricep and bicep muscles. Now, let us see the ultrastructure of the muscle fiber. Now, as we have just now mentioned that the muscles are made up of the many muscle fibers. Now, these muscle fibers, if you see, they are covered by a membrane known as the plasma lemma of the membrane. Here in case of the muscle, rather it can be called as the sarcolemma. Otherwise, in general we call it as the plasma lemma. In case of the muscle, the plasma lemma covering the cell, we call it as the sarcolemma. So, this sarcolemma which is covering the muscle fiber is connected to a complex system which is known as the T system, that is transverse tubules. Now, these T system is nothing but the invagination of the sarcolemma inside the muscle fiber which runs across the muscle fiber. Now, apart from this, we also have the sarcoplasmic reticulum present in case of this muscle fibers. Now, this sarcoplasmic reticulum, it plays very important role in the muscle contraction because the calcium which is very important in the muscle contraction will be present in the sarcoplasmic reticulum. Now, let us see the ultrastructure of the muscle fibril. The cytoplasm of the muscle fiber is known as the sarcoplasm. inside the sarcoplasm we find many cells, they are known as the myofibrils. Now, let us see the ultrastructure of the myofibril. Now, under electron microscope, if we see the myofibril, we find that they are different types of bands, that is a thick band and thin band alternating with each other. Now, this is the thick band and this is the thin band, we find they are alternating with each other. Now, in fact, the striations which are seen on the muscle fiber are because of the presence of these bands. Now, the thick filament which is known as the A band and the thin filament is known as the I band. This is because the A band is anisotropic and birefringent and the I band is isotropic. Now, in the center of this I band, we also see a line dividing the I band into two parts. This line we call it as the Z line. In fact, this Z line is derived from a German word which is known as the Zwischenschreiber. Zwischenschreiber means it is just a line drawn in the center, that is a German word. So, it has been derived from the German word. So, it is known as the Z line. So, the presence of this Z line in the center of the I band divides the I band into two parts. So, the space in between this Z line and another Z line forms a unit. Now, this unit in case of muscle fibril, we call it as a sarcomere. That means, muscle fibril is made up of different units known as the sarcomeres. Now, similarly, in the center of the A band, we see a clear zone which is known as the H zone. Now, in the center of the H zone, a line which is present is known as the M line. Now, another interesting thing we find is, now these filaments, that is the A band and I band, they are also linked by the presence of cross bridges. Now, if we take the section of these filaments, we can see that now the section taken in the H zone, we find all the thick filaments present, whereas the section taken in the I band, we see all the thin filaments present. Now, in case of the A band, we see both thin and thick filaments present. That means, each thick filament is surrounded by 6 thin filaments and each thin filament is surrounded by 3 thick filaments. Actually, morphologically speaking, there are 2 types of filaments, that is the thick filament and thin filament, which we were just now discussing. Now, in fact, this thick filament is present in the A band and the thin filament is present in the I band. To have an idea about the relative dimensions of these bands, the A band measures 1.5 micrometers long and 10 nanometers diameter, and I band measures 1 micrometers long and 0.5 nanometers in diameter. Now, in a living muscle, the interesting observations were made that during contraction, the length of the A band is seen constant, whereas the I band varies. That means, the I band is seen rather reducing the length, whereas the A band was constant. This is because of the sliding of the I bands into the array of the A bands towards the H zone during contraction. That is the reason why not only the I bands are pulled towards the H zone of the thick filament, with the result that the sarcomere also is becoming short and shorter. That means, during contraction, the length of the sarcomere also is seen contracted. Now, molecular organization of the contractile system. Now, if you see the molecular organization, it includes the following proteins such as the force generating proteins example are the actin and myosin, and regulatory proteins, examples tropomyosin and troponin, and the structural proteins alpha actin, M band proteins and C proteins. Now, these force generating proteins, if we see the structure of this actin and myosin, the myosin in fact it is a very large protein. The myosin itself forms the 50% of the muscle protein. Now, the molecular weight is 500,000 Daltons, and it comprises of 2 heavy polypeptide chains having 200,000 Daltons and 4 small subunits. Now, these 2 polypeptide chains, they form helix, they twist around each other and forming a helix. Now, the entire myosin molecule can be divided into 2 parts. One is LMM, that is light meromyosin, and HMM is heavy meromyosin. In fact, the light meromyosin is a straight chain, that is a helix. Now, coming to the HMM, this HMM or heavy meromyosin can be still further divided into 2 parts, that is HMM S2 region, heavy meromyosin H2 and heavy meromyosin S1. Again the HMM S2 is a straight helix, but the HMM S1 we find the globular structures. In fact, one of the ends of this myosin molecule, this forms a globular structure which in turn forms the cross bridge. Now, this part is very important because the myosin ATPase lies here, and this is in fact the active site in getting attached to the actin filament. Now, of course, we have already seen that this myosin molecule is present in the A band, that is thick myofilament. Now, coming to the thin myofilament, that is I band, that is made up of different proteins such as actin, tropomyosin and troponin. Now, this actin here, it is the globular actin. Now, all these globules of the actin, they form a chain, and the 2 chains again twist each other in forming a helix. Now, this tropomyosin, which is filamentous in structure, this tropomyosin will be present in the grooves of the twisted actin. At the end of this tropomyosin, that means at one side of this tropomyosin, the troponin is attached. Now, the troponin is almost, as you are able to see here, it is oval in structure, but still the troponin is not that simple. It in turn, it is a complex of proteins, that is it has 3 parts. One is troponin I, troponin C and troponin T. Troponin T means this part of the troponin, this will have a high affinity for tropomyosin. This part of the troponin gets attached to the tropomyosin, and this troponin, that is troponin C, this will have the affinity for the calcium molecules. The calcium molecules are very important in the process of the muscle contraction. So, these calcium molecules, when they are released from the sarcoplasmic reticulum, they are attached to this part of the troponin, that we call it as the troponin C. Now, this is the troponin I, that is this part inhibits the myosin ATPase activity. So, these are the very important proteins we come across in the muscle. Now, let us see the mechanism of muscle contraction. Now, this mechanism of muscle contraction can be explained by taking a very widely accepted model, that is sliding filament theory, which is proposed by A.E. Huxley and Hansen in 1954. Now, here what happens is, at the time of contraction, we see that the length of the A band and I band remains constant. The length of the A band and I band also, they remain constant. But the I band, it appears to be short because the I band slides in the area of the thick filament, that is the H zone of the thick filament. Hence, what happens is the length appears to be short, but in fact the length of the I band also will not change. Now, with the result, because the I bands are pushed inside the A bands, with the result what happens is the length of the sarcomere also reduces. Another very important point is these A band and I band, they are attached to each other by the presence of the cross bridges. Now, these cross bridges are formed by the thick filament, that is A band, that means by the myosin heads. So, from the thick filament, from the myosin, this part of the myosin forms a cross bridges and then it gets attached to the actin filament. Now, these actin filaments are again pulled inside. With the result, what happens? If the intensity of the contraction is more, then at times the H zone, it's gradually it becomes smaller and smaller. At one time, if the contraction is really very heavy, then the H zone completely disappears. Another very interesting observation is made that these cross bridges, once formed from the A band and then attached to the I band, during the process of contraction, they do not remain at one side. In fact, these cross bridges attaching the I bands, they attach and detach, reattach the next side. Thus, they pull the I bands towards the H zone. So, these are the very important items which we find during the process of muscle contraction. Now, we will have to study about the energetics of the muscle contraction also, because now we have seen that during contraction there is so many things are happening. The filaments are pulled inside and the cross bridges are formed. So many things are happening. For this, definitely the muscle requires some energy. So, let us see how this energy is formed, where from the muscle is getting energy for the muscle contraction. So, coming to the energetics of the muscle contraction. Now, ATP is the very immediate source of the muscle contraction. In sense, ATP is the immediate source of the energy required for the muscle contraction. Now, this ATP, adenosine triphosphate, in the presence of calcium ions and ATPase enzyme, it splits into ADP and inorganic phosphate, at the same time releases energy required for the contraction. Now, in case of the muscle, the immediate ATP is generated by the energy rich compounds such as creatine phosphate in case of the vertebrates and arginine phosphate in case of the invertebrates. Now, these energy rich phosphates known as the phosphogens. So, this creatine phosphate, in fact it rephosphorylates ADP into ATP. That means, it is a reversible reaction. Now, here this creatine phosphate, immediately the ADP which are produced here will be phosphorylated to ATP. So, that again the ATP molecules are available in the muscle for the contraction. Okay. So, the creatine phosphate plus ADP gives creatine plus ATP. So, apart from this, we all know that in the body with the glycolysis and oxidative phosphorylation, we have many number of ATP molecules. Now, so we have seen this energetics of the muscle contraction. Let us also see about the initiation of the muscle contraction. Now, how exactly the muscle contraction initiates? Now, all these muscles are associated with the nerve endings. So, the nerve impulse actually that initiates the muscle contraction. Now, when the muscle is stimulated, immediately the impulse is carried and it depolarizes the sarcolemma and it propagates so rapidly that entire membrane of the myofibril is depolarized. That means it gets depolarized and also the sarcolemma and also the transverse tubular system, that is the T system, also gets depolarized. So, once this system gets depolarized, in fact this depolarization we also call it as a excitation. That means once it gets excited, what happens is from the sarcoplasmic reticulum the calcium ions are released. So, these calcium ions are very important because we have seen that only in the presence of calcium ions, here also we have seen that the ATP is giving out the energy. So, the calcium ions are very important in case of the muscle contraction. So, only after it gets depolarized from the sarcoplasmic reticulum, these calcium ions are released. So, once the calcium ions are released, they get attached to the troponin, and then once the troponin is attached to the calcium ions, then there will be a structural configuration change in the structural configuration of tropomyosin and troponin, thus exhibiting the active site of the actin for the myosin cross bridges gets attached. That means in the presence of a calcium only all these changes are taking place. So, like that the cross bridges gets attached to the I band, okay, the thin filament. So, once the calcium is released or the amount of calcium is depleted, then what happens? The calcium is immediately released from the troponin filament and then the relaxation takes place. Now, another interesting thing here we can mention is the rigor mortis, which you must have heard that we find in case of the dead. The rigor mortis is nothing but the detachment of the actin and myosin filaments does not take place because there will not be any production of ATP molecules. So, and then the production of ATP molecules is not present means again the process is not happening, that is the calcium ions are not released. So, what happens? The actin and myosin filaments will not get detached in case of the dead person, that is what we call it as the rigor mortis. So, so far you have studied about the muscle and the structure of muscles and different types of muscles and the process of muscle contraction and the energy required for the muscle contraction etc. in this episode.